Hey everybody, welcome to video notes for topic 8.12 and 8.13. We're talking about dose response studies and lethal dose 50 or LD50. So these are studies that expose plants or animals to different amounts or concentrations of a chemical and observe the effects it has on the organism. So in this case, the independent variable is the concentration or the dose of the chemical. So we might measure that in the number of milligrams per kilogram of body weight of that organism, or we could say the number of milliliters per liter of a pesticide in a fish tank or something like that. Then the dependent variable is going to be the effect that's measured on the organisms, and it's most commonly death. So sadly, the dependent variable for these studies is most commonly the percent mortality, or the percentage of the organisms that die at the different concentrations. Now sometimes sublethal effects are measured. Sublethal means non-lethal. It does not kill them, but it has a harmful effect, such as causing infertility or nerve damage. And so in these studies, the chemical could be added to the food, air, or water, in which case we would call it a concentration. So we could say the concentration of NOx in the air. And in the case of giving it to the organism directly, having them absorb it or ingesting it, that would be called a dose. So again, that might be the number of milligrams per kilogram of body weight that they ingest or take in of that chemical. Now we'll talk about a dose-response curve. So the data from a dose-response study are graphed in a dose-response curve. And on the y-axis, we have the dependent variable, which again is percent mortality, the number of the organisms that die. And on the x-axis, we have the independent variable, the concentration or the dose of the chemical. So remember, this is kind of typical in graphs that are showing data from experiments. We have the dependent variable, or the outcome we're measuring, on the y-axis, and we have the independent variable, or what we're changing, on the x-axis. And they typically, dose-response curves, have an S shape, meaning there's very little mortality at low concentrations. And the one I drew was not nearly as accurate as the one that's in your book. The one that's in your book is far more accurate. It shows that there's actually 0% mortality at low concentrations, and it takes a while before we actually reach what's called the threshold. So the threshold, which might be right around 3 of this kind of hypothetical dosage, because that's the first point, the first dose where the effect is actually detected, meaning we're measuring some mortality. So even though only 1 or 2% of the organisms have died here, that would be the threshold, because it is the point where we do start to detect mortality. Now typically they have this S shape because once we get over that threshold, then organisms really start to die at high rates, because we are in a territory that's far more toxic, far more outside their range of tolerance. And then as we get to the top, most of the organisms have died, and the curve levels off because these organisms are probably, have a genetic mutation or have some sort of tolerance to it. And so then it really takes a lot to kill up to 100%. What you'll notice here, and we'll just slide over a little bit, is this dotted line drawn in at the 50% mortality. And this measures the dose or concentration at which 50% of the population of this particular species are dying. Sometimes we call this 50% mortality. So in this case, it's maybe 5.5. So, the LD50 would fall somewhere between 4 and 6, probably close to 5.5, because that's where 50% of these organisms are dying. You can also measure the sublethal effect. So sometimes studies will look at the percent of organisms that develop a certain type of cancer, or that develop nerve impairment or infertility. And in this case, we would call that ED50. So ED50 represents a sublethal effect happening in 50% of the organisms. And it just depends on the study. So again, LD50 though stands for lethal dose 50%. It's where 50% mortality happens, and you find it by finding the point where 50% mortality matches up with the dosage or the concentration of the chemical. Now we'll talk about how this LD50 value is actually used. So in dose-response studies, we're never testing on humans. That's unethical. That's an important point here. So instead of testing on humans, we use models. These are organisms that function similarly to other organisms in their large classifications. So we use rats and mice as a mammal model, meaning we extrapolate or apply the results of rats and mice to all mammal organisms. We use fish for aquatic, amphibian, and reptiles, and we use birds for land or terrestrial amphibians and reptiles. What we do with the LD50 level that we discover, which in this case would be for this pesticide that's being given to rats, we've determined that the LD50 level is 2 milligrams per kilogram of body weight in that rat, in body mass. So the way that we apply this to humans is dividing it by 1,000. So we cut this down by 1,000, divide it by 1,000, and that's going to give us the safe level for humans. Another way to think about this is dividing it by 10 3 different times. So I have this math written in pencil down here for an example of how to do this. We take the LD50 level in rats, we divide it by 10, because we kind of assume, well, if 2 milligrams per kilogram kills half of them, dividing that by 10, a tenth of that, should be relatively safe for rats. We divide it by 10 again to reflect the fact that rats may be less sensitive to this chemical than humans are. And then we basically divide it by 10 one more time to just kind of have what's called an abundance of caution, to be totally safe, to be precautionary, rather be safe than sorry. So ultimately for humans, 0.002 milligrams per kilogram of body mass is what we would consider safe for us to have in our bodies. And so again, this is an example of how we do a dose-response study for a certain pesticide using rats as a model. Finally, we have to talk about some limits of these studies. So dose-response studies are considered acute, which means that they're short-term studies. They have limitations. So since the organisms are only modeled for a short period of time, we don't really see longer-term effects. We only see immediate effects. It's also limited to lab conditions in a test population. So we don't know how this would truly impact wild populations. We don't know how it would impact the ecosystem as a whole. We don't know if it would affect their predators and their prey. So there's huge questions that remain even after doing an LD50 study. Chronic studies do allow us to follow the organism for longer, such as following fish from the time that they hatch to sexual maturity to see if there's problems with their reproductive systems. So this is one potential solution, is doing what's called a chronic or long-term study.